Thank you so much for inviting me to this symposium. And today I want to tell you a little bit about work we're doing in my lab to look at how partially occluded objects are processed in the primate brain. So, when we look at a complex visual scene such as this one, our visual system effortlessly segments the scene and we're able to recognize a variety of objects. Our visual system is remarkable at this sort of scene understanding. We can interpret scenes even when they are impoverished. For example, with this scene, even though at first we are unable to parse this, some of us may be unable to parse this visual scene. All we need is a little bit of priming, and we should be able to see this Dalmatian dog effortlessly from here on out. So, this is one shot learning. Um, One of my all-time favorite visual illusions is the Bregman's bee illusion, both for its simplicity and its power in revealing the importance of context in parsing visual scenes. These yellow bits don't provide a unified percept, but if I were to add an appropriately positioned ink stain, then the image then coheres into a set of partially occluded beads. Now, studies over decades have shown that this sort of parsing visual scenes is based on processing information along the ventral visual pathway, which runs from area V1 in the back of the brain to V2, V4, and then subregions of the inferior temporal cortex. In the first stage along this pathway in area V1, we know that based on studies for decades, we know that neurons encode information in terms of local orientation and spatial frequency information. And we have detailed models for how this sort of representation is built from signals earlier in the visual system. As we go beyond V1 into V2, V4, and the subregions of IT cortex, we know that receptive field sizes increase in size and response properties also become much more, response selectivities become much more complex. For example, in the highest stages along this pathway in IT cortex, we know that neurons are sensitive to stimuli like faces, hands, etc. But little else is known about how the computations arise, how neurons are arranged, and how they function. So, the major focus of the research in my lab is area V4, which is an intermediate stage along this pathway. We're interested in trying to understand how the basis of representation in area V4, what are the underlying computations that give rise to these representations, what is the functional organization? So, how are the different response properties arranged across the sheet of cortex? And we're also interested in understanding what are the network interactions between B4 and other brain regions, and also the population response dynamics to underlie our ability to recognize parse scenes and recognize objects. So today I'll be talking about the last two of these, and specifically in the context of processing partial occlusion. So the question before us today is to understand how do visual and frontal cortex interactions support our ability to recognize partial occluded objects. Um, when we, you know, Partial occlusions are all around us. To begin to understand how this is, but our visual system is remarkable at recognizing objects despite occlusions. And to begin to understand how recognition of partial occluded objects is achieved, we uh, we studied the responses of neurons in area V4 under different levels of occlusion. So we started with a set of shape stimuli, and then we presented the stimuli either in isolation or with a randomly positioned occluding dots superimposed on the stimulus. And we titrated the level of occlusion by varying the diameter of these dots. So down here, the shape is much more occluded than up here. And we quantified the level of occlusion by computing the fraction of unoccluded area. So when it's 100% unoccluded, when there are no dots at all, it's 100% unoccluded. And then as the diameter of the dots increases, this number goes down. So when this number is high, it means the stimulus is less occluded. And when the number is low, it means it's more occluded. We then studied the responses of many B4 neurons to these kinds of stimuli. And I'm showing you the results from one example neuron to this particular shape stimulus. The stimulus comes on at time 0 and goes off at time 600 milliseconds. And I'm showing you the firing rate of the neuron, the spikes per second. Now, in black is the responses of the neuron to the unoccluded stimulus. And so you can see there's a transient portion of the response, and then there's a sustained portion of the response, and the response goes down back to baseline. Now, as I add more and more occluders, the response curves are drawn in blue, then yellow, and down to red. And you can see that the responses, the initial transient responses, decline with increasing levels of occlusion. And this is what we saw in lots and lots of people neurons. And this is also consistent with what other people have shown in IT cortex. For example, Kovacs and colleagues showed that the responses of IT neurons declined. So here they're showing the response trend on the y-axis to rank ordered set of stimuli. The line in black is the responses to the unoccluded stimulus, and you can see a nice shape selectivity for this group of neurons. And as you increase the level of occlusion, the magnitude of the response goes down, but the shape selectivity remains consistent. So this is what we found in B4 as well. And this is also what people have found in human cortex. So based on intracranial field potentials in occipitotemporal cortex in epilepsy patients. So then there's a conundrum, right? So responses in B4 and IT decline with increasing levels of occlusion, but we're still able to recognize these shapes. So what gives? So how are we able to recognize these partially occluded shapes? One hypothesis, based on lots of speculation, let's say, is that there might be feedback from higher cortical areas. In this case, we are thinking of prefrontal cortex back down to visual cortex, specifically area B4, that might support our ability to recognize in these difficult sort of situations. And there are lots of reasons to motivate prefrontal cortex as the source of this feedback. PFC is thought to be important for executive control of behavior. It's implicated in the maintenance of working memory. And past studies have also shown that prefrontal cortex is involved in recognition, and it interacts with regions in the visual cortex, specifically area visual. So to study prefrontal cortex, we then set out by designing a sequential shape discrimination task. And the sequence of events in that task are shown in this slide. So each trial begins with the presentation of a fixation spot, followed by the presentation of a reference stimulus at central fixation. And then after a delay, a test stimulus is presented in peripheral location. And we designed it so that this way, because we wanted to present the stimulus in the receptive field of the V4 neurons as well. And then the task of the animal was to report whether these two shapes are the same or different. If they were the same, the animals had to saccade to make a rightward saccade. If they are different, they have to make a leftward saccade. Now, this test stimulus was partially occluded to titrate the levels of occlusion. We used the same strategy as I described before. So randomly positioned dots with varying diameter of dots. Animals performed this task really well when the partial occlusion levels were low, and when the occlusion levels were higher, the task became more difficult for the animals. But I won't tell you much about the performance of the animals for today's talk. We then studied the responses of neurons in V4, in prefrontal cortex, and also in IT cortex. And we did these experiments sequentially with the same pair of animals. So I've already told you what we see in V4. We see responses declining and shape selectivity declining with increasing levels of occlusion. So I'm going to start by showing you some responses in prefrontal cortex, because what we found there was quite surprising. So these are the responses of one example neuron in prefrontal cortex. Again, color schemes are the same as what I showed you. 0 again is when the stimulus comes on, and 600 is when the stimulus goes off. And what you can see here is distinctly different than what I showed you in V4. So the responses to the unoccluded stimulus, here again shown in black, are weak, and the responses are much stronger to occluded stimuli. And this is the case for both of the shapes that we used in this particular session. And the responses are stronger for unoccluded stimulus, but for the occluded stimuli, but also shape selective. So responses are stronger to this particular stimulus than this particular stimulus. And this is what we saw across the population of prefrontal cortical neurons. So here I'm showing you the average normalized response of a population of prefrontal cortex neurons. Again, in black is the responses to the unoccluded stimulus, and the different levels of occlusion are in different colors. And so, again, the responses are weaker for unoccluded as compared to the occluded. And here on the right panel, I'm showing you shape selectivity. And you can see, again, shape selectivity is weak for the unoccluded stimulus and stronger for the occluded stimuli. So what we're seeing in prefrontal cortex is stronger responses and stronger shape selectivity with for occluded stimuli. Okay. Now, next, we wanted to look at the timing of the prefrontal cortical responses, and here I'm showing you the peak latency, the distribution of the peak latency across prefrontal cortex, and the median of this distribution is around 150 milliseconds. So, if we want to look for influence of feedback from prefrontal cortex, we might want to look at this time point around 150 milliseconds in the V4 responses. So, as I said, we'd already done all of these experiments in V4 with this exact same behavioral task and with the same monkeys. So, we went back to our V4 responses and looked at the dynamics. We analyzed the dynamics of V4 responses to see if we can find any influence of feedback in our V4 data set. So I'm showing you an example V4 neuron that shows no influence of feedback. Essentially, the influence of occlusion is very consistent across time for this particular neuron. So again, the responses are strongest for the unoccluded stimulus, and the responses to the different levels of occlusion are just weaker versions of that same response. So if I wanted to predict the responses to these different levels of occlusion, all I need to do is take one constant multiplicative factor that doesn't change across time to predict responses to these different levels of occlusion. In fact, that's what we did. We predicted the responses of the neuron as a function of occlusion level and time by simply multiplying the response to the unoccluded stimulus by a factor which depends just on occlusion level. So the most parsimonious explanation is that we don't have to invoke any sort of feedback that modifies the response at a later point in time. This was roughly 60% of the neurons that we recorded from in V4. But the remaining 40% of the neurons showed some very interesting dynamics. And an example of that is shown in this next slide. So this is a neuron that had two dynamic peaks, two transient peaks in its response. So the first transient peak that's marked by this black bar here showed the standard response profile, where the responses are strongest to the unoccluded stimulus and decline with increasing levels of occlusion. When you look at the second peak, the responses are actually stronger for intermediate levels of occlusion. So if we were to plot the responses during the first and the second peak as a function of occlusion level, which is what I'm showing you here. So x-axis is the level of occlusion, y-axis is the response during these two time periods. For the unoccluded response, for the response during the first transient peak, responses are strongest for the unoccluded stimulus, and they decline with increasing levels of occlusion. For the second transient peak, however, you have an inverted U function where the responses are strongest for the intermediate levels of occlusion. Here are two other examples, again, showing two transient peaks. Again, during the first transient peak, response is strongest for the unoccluded stimulus. That is definitely not the case for the second peak. The response is stronger for intermediate levels of occlusion for during the second peak. Now, we then looked at the timing of the peaks and compared it with that in prefrontal cortex. So I showed you this distribution already, and the median of this prefrontal cortex distribution is around 150 milliseconds. Now, if we compare that with the timing of the first peak and second peak in B4, the peak of prefrontal cortex lies somewhere in between these two peaks, making it possible that the second transient peak is a direct result of feedback from prefrontal cortex. Um, so, so the response of, as I said, response of most prefrontal cortex neurons is timed in between these two peaks. Now, so we got really excited about this. All this is based on not direct simultaneous recordings, but we could construct a toy model based on these results to explain the observed results on the basis of a V4 PFC interactive model. So I'll walk through that model very briefly here. So basically, we started out with a pair of V4 neurons with different shape selectivity, and if these two neurons are driven by as a partially occluded stimulus, then you can model the responses where the responses you have an initial transient and a sustained response, which declines with increasing levels of occlusion. Now, we send a feed forward input from that to prefrontal cortex. Prefrontal cortex also receives a second input, which is characterized by the level of occlusion. Here, the level of occlusion, the responses, this gain modulation is inversely proportional to it's directly proportional to the level of occlusion. So it is higher for high levels of occlusion and lower for low levels of occlusion. So inversely proportional to the unoccluded area, directly proportional to the level of occlusion. Then you get the characteristic responses that we saw in prefrontal cortex, where the responses are weaker for the unoccluded stimulus and stronger for higher levels of occlusion. Now, if we were to send a feedback from prefrontal cortex back to V4 with some synaptic adaptation, then you can simulate this second bump, that second transient peak that we saw in V4 responses, which is stronger for intermediate levels of occlusion. Now, this is all based on our V4 and prefrontal cortex recordings. We were also interested in understanding what happens in IT cortex. And I'm going to summarize that with just one slide here. It's all published. But basically, in IT cortex, we saw two kinds of responses. The first one is very similar to what we saw in V4, where the responses were strongest to unoccluded stimuli, weaker for higher levels of occlusion, and these neurons are highly shape selective. This was roughly 40% of the neurons in IT cortex. Second group of neurons showed stronger responses to occluded stimuli, similar to prefrontal cortex, but these neurons were not shape selective, so they were explicitly sensitive to the level of occlusion, and this was roughly 60% of IT neurons. So based on this, we modified our feedback model where V4 and IT sent feed-forward input into prefrontal cortex. The V4 and a subgroup of IT neurons carry information about shape. A subgroup of IT neurons carry information about occlusion. And these signals together modulate PFC responses, which then provides a feedback back into V4. Okay, so this is the basic model, hypothetical model that we were working with. But as I said, all of this is based on sequential recording of V4 and prefrontal cortex. We did not directly simultaneously record activity from both areas. So that is the experiment we're working on currently right now. So we're perturbing the activity of prefrontal cortex neurons and then asking how is that influencing the responses of V4 neurons. So the basic logic here is that if we suppress the activity of prefrontal cortical neurons, the question is, how does that influence the second dynamic peak in area B4. And to address this question, we've now developed a cortical cooling probe, which can be positioned on top of prefrontal cortex and which can cool the underlying cortex by passing chilled ethanol through the probe. We can monitor the temperature of the probe. We also have slits cut through the probe so that we can pass neuropixel probe or thermocouples through it to measure the temperature or the neural activity of underlying cortex. We've completed these experiments with our first monkey, and we're working on the experiments with our second animal. So I can give you a very quick version of the results so far. So let me show you first the titration of temperature of the cooling probe. So the probe temperature, we can modulate all the way from 30 to 10 degrees centigrade or so. But the brain temperature modulates over a much smaller range. And that's good because we don't want to shut down the activity of neurons. We really want to perturb the activity of the neurons mildly to see how that affects the responses of B4 neurons. As we're doing this experiment, we can study the responses of neurons in prefrontal cortex. We're using neuropixel probes now, so we can observe the activity across all the layers of cortex. I'm showing you up here the temperature of the probe and the activity, the correlated activity of the neurons down below, and we can see there are two things that we can observe here. We do see the influence of cooling across all the layers of cortex, which is good. Um, we see some neurons that show a positive correlation. So the responses are suppressed during the cooling phase, which is shown here in blue, and then stronger during the warm phase. But there are other neurons that respond only during cooling. Their responses are suppressed during the warm phase. And so we can characterize this by computing the correlation between the temperature and the neuronal activity. And across the prefrontal cortical population, we see the vast majority of neurons are actually positively correlated. So that means their responses are suppressed during cooling, and there are a subset of neurons that are negatively correlated as well. We can look at whether there is any difference in spread across the layers, and we haven't seen anything. But we're also analyzing how that relates to waveform shape to see if we can relate this positive and negative correlation with potential types of neurons. So far, we just have preliminary results on this, and it looks as though neurons associated with narrower waveforms are associated with a negative correlation, more of a negative correlation in prefrontal cortex. The big question is whether suppressing prefrontal cortex activity actually influences B4. And our original hypothesis was that suppressing PFC would also suppress B4. And that's not exactly what we find. So this is recordings from B4. Again, this is the probe temperature. These are activity traces. The effect is not as strong as in prefrontal cortex, which makes sense. It's not a direct effect. And again, we see all kinds of modulations. So, for example, down here, we see a neuron that actually is enhanced during cooling. We see neurons that are suppressed during cooling. And if we were to look again at the correlation, we actually find that a greater proportion of B4 neurons activity actually increases with cooling. Okay, so it's 1.5 times more neurons have an enhanced activity during cooling. And we're still working on trying to relate that with the shape of the waveforms. So then this blows our hypothesis a little bit because we were imagining that it was an excitatory feedback from prefrontal cortex that's modulating V4 activity. But now we're thinking about it, thinking about disinhibition control as a possible alternative model where prefrontal cortex input actually modulates whether V4 is more in a recurrent mode or a feed forward mode. So the idea comes from rodent studies where they've shown that the activity and excitatory feedback can go through a sequence of two inhibitory uh, neurons to then disinhibit the circuit. And here we're thinking about how such a circuit might make the system work, alter the weights of the system such that it operates more in a recurrent mode or a feed forward mode. So to unpack that a little bit, if your prefrontal cortex activity is low, then V4 neurons may be in more of a feed forward mode. But if the prefrontal cortex activity is high, then the circuit might be in more of a recurrent mode. But this again is a speculative model. We're still trying to analyze our data and trying to understand if we can sort of relate the various waveforms to the kind of correlation that we see based on the modulation of prefrontal cortical activity. So with that, let me summarize. So we hopefully have a few minutes for questions. So we think that dynamical interactions between visual and frontal cortex may be important for robust representation and recognition under occlusion. We're using reversible perturbation to try and dissect the interactions between V4 and PFC. We think, based on our preliminary data, we think that it may not be a straightforward excitatory feedback relationship. It might be more of a disinhibition control model, but still the jury is still out on that. And we think these sort of dissections might be important for us to understand how diminished sensory frontal communication might be important for sensory symptoms in some sort of brain disorders. With that, I want to acknowledge all the people who've done this work and our funders, and I will take some questions.
Fantastic. Such a beautiful, primate work takes so much effort and beautiful illuminating results. So time for questions. Anybody like to raise their hand for the questions? And may I ask a first question regarding whether do you think this prefrontal activities may be task dependent? So if your monkeys are not trained for this task, will they still be more activated by occlusion than non-occlusion?
That's a great question. I don't think we know the answer to that. I want to believe that it's very much dependent on learning and the need to get juice, right? They're getting rewarded doing this task. Mm-hmm. And so I think a lot of this is I think of prefrontal cortex as an adaptive network that learns to maximize reward, for example, in this situation. So I suspect that it may not, prefrontal cortex neurons may not be active if the animal was not trained on this task.
Ooh, yeah. And like IT looks like it's in between, yeah? So as if maybe if one is task dependent, maybe IT also, of course, this is, very interesting. So yes, please, anybody wants to raise your hand and ask questions?
There's one question in the chat.
Oh, there's one question in the chat. Okay, the question is, would the symmetry of the occlusion stimuli play a role in the detection of the occluded stimulus, assuming that the visual system is biased towards a symmetrical stimulus against asymmetric occlusions? This question is by Wakia Khan.
Yeah, that's a good question. We, a lot of the tasks that we did, we used only these occluding dots in our experiment. We haven't used any other forms of occlusion where you might see the influence of asymmetry or symmetry on the responses. So our experiment is very symmetric in the fact that these occluders are all circles and they're all randomly positioned, and we varied only the area of the occlusion. So it's a very we haven't explored the space much, but I believe symmetry and the animal's familiarity with the occluders is also probably key in the performance of the staff. Certainly.
And you are, in addition to the occlusion stimuli, how about the occluded stimuli? And your objects are not necessarily all bilateral symmetric, the occluded ones, no?
Yes, they're not all bilaterally symmetric, but there is some symmetry in the features of it. So symmetry as in a high level symmetry, as in, they are all contours, close contours that are two-dimensional shapes that, that are flat silhouettes. There is nothing diverse about them. Yes, but they're not bilaterally symmetric. That's correct.
Well, thank you so much. It's so fascinating. This is like the last talk of the symposium, and I like to, in fact, it's still open, so people can ask questions, continue if the speakers are still around. But first of all, let me just thank all the speakers and presenters for this fascinating treat. Well, actually, there is a question. Let me cut to Tom, who's asking this question. Yeah, because Tom is another V4 neurophysiologist, so another expert. So Tom, please go ahead. Did I manage to? Yeah, please go ahead.
Thank you. Great talk. I was wondering about the occluder signals that you showed. So it seemed that your data suggests that the information of the occluders is present in IT, but not in V4. So do you think that information is computed in IT independent from any V4 input to IT, or how do you think about that?
Yeah, so we did the experiments a bit different between IT and V4. And I think that may be the source of this difference. I don't believe that that the signal is not there in V4. So in V4, we did the series. The way we did these experiments was we started with V4, and then we went to prefrontal cortex, and then we did it in IT cortex. So things varied over the years how we did these experiments. In V4, we put in an electrode, we mapped the receptive field. We characterized the shape tuning preferences of the neurons. We picked a preferred shape and a non-preferred shape, and then we did the experiment. So if the neurons didn't show much shape selectivity, we moved on. We found another neuron. So these were all sort of customized for neurons that showed some basic selectivity amongst the set of shapes we were using. In both in prefrontal cortex and in IT cortex, we used multi-contact electrode probes. And so we did not customize the experiment to the properties of the neuron. We just, we put the probe in, we took whatever neurons we got, and we just picked two random shapes and we proceeded with our experiments. And so I think our prefrontal cortex and IT recordings are more representative of the general population, subpopulation of neurons. And so we saw that there were neurons that were sensitive to both aspects. In V4, we've done other experiments since with other kinds of stimuli, and we see that neurons are sensitive both to shape and to texture. So we suspect that, if you think of those occluders as a texture pattern, then certainly you'd see many neurons in V4 that would be sensitive to that stimulus as well. Yeah. Thank you for that question.